واللي هي استكمال للبلازما ممبرين ريسبتورز. Okay. In the last lecture, we studied the second messengers, cyclic AMP and cyclic GMP. So in the last lecture, we studied cyclic AMP, cyclic GMP, and we said we will continue in today's lecture with the rest of second messengers. In today's lecture, we will continue with IB3, DAG, diacylglycerol, and calcium ions. So in today's lecture, we will continue in IB3 and DAG. We will see their synthesis pathway for IB3 and DAG. They have a similar synthesis pathway. This is the cyclic correlation. Okay. Phosphatidylindole. So now we will study the synthesis pathway for IB3 and DAG. They have similar synthesis pathway nearly. Phosphatidylindole. What is the phosphatidylindole? Phosphatidylindole, BI, shortly known as, is a cell membrane phospholipid. So, is a phospholipid found in the cell membrane. The cell uses this phospholipid to synthesize IB3 and DAG, as we will see in this slide, the synthesis pathway. So, phosphatidylindole is a protein where it is found in the cell membrane. Phospholipid found in the cell membrane. From phosphatidylindole, the cell will synthesize, as you can see, IB3 and DAG, the diacylglycerol. The first step in their synthesis is phosphorylation of phosphatidylindole using 2-ATP. So the first step is phosphorylation of phosphatidylindole using 2-ATP, thus converting phosphatidylindole into phosphatidylindole 4,5-biphosphate. Means there are two phosphate bonded to which atoms? Number 4 and 5. This is why we call it bi-2-phosphate, shortly known as BIB2. So the first step is the phosphorylation of phosphatidylindole using 2-ATP. Thus, phosphatidylindole is converted to BIB2. Then phospholipase C. Phospholipase C, as we will see, just remember this name, is an effector protein found in the cell membrane. As of adenylyl cyclase, guanylyl cyclase, and phospholipase C also is an effector protein found in the cell membrane, and its function is the synthesis of DAG and IB3. So phospholipase then cleave BIB2 into DAG and IB3. As you can see, it's very simple synthesis pathway. So phosphatidylindole using 2-ATP is phosphorylated to BIB2. Then phospholipase C cleave BIB2 into IB3 and DAG. As you know, diacylglycerol is hydrophobic. The fat tissue, the adipose tissue in our body, it has triacylglycerol. A glycerol attached to it 3 fatty acids, while diacylglycerol is a glycerol but attached with it 2 fatty acids. Because it is hydrophobic, it remains in the cell membrane and moves until it finds protein kinase C and activate it, as we will see in the figure next slide. While IB3, so what is the function of DAG? Activation of protein kinase C. What is the function of IB3? Release of intracellular calcium ions, normally found in the endoplasmic reticulum. See, calcium ions also their function is they can also activate protein kinase C. So protein kinase C can be activated by DAG and calcium ions. And calcium ions, it regulates other enzymes as we'll see. And protein kinase, because it's kinase, it phosphorylates different enzymes and proteins. This is the synthesis pathway for DAG and IB3. See here. And just remember what we said in the last lecture. This is the G protein coupled receptors, and this is the G protein. When the hormone, same G protein coupled receptors. What is the difference between one hormone and the other is the effector protein here. In the last lecture, we said that the effector protein adenylyl cyclase, thus it produces cyclic A and B. Then the second example was guanylyl cyclase, thus it produces the second messenger cyclic G and B. While here, what is the effector protein? Phospholipase C, that will cleave, as we said, BIB2 into DAG and IB3. So just to repeat what we said in the last lecture, as all G protein coupled receptors, once the hormone binds to G protein coupled receptors, what happened? Conformational change that leads to the activation of G protein. Then the replacement of JDB with JTB on the alpha subunit. Then alpha subunit is separated from beta and gamma. It moves along the cell membrane until it finds what? An effector protein. What is the effector protein here? Phospholipase C. Phospholipase C, as we said a minute ago, what is the function of phospholipase C? It cleaves BIB2 into DAG and IB3. As we said, DAG is hydrophobic. It can't diffuse in the cytoplasm, so it moves along the cell membrane until it finds protein kinase C and activate it. While DAG, while IB3 is hydrophilic. IB3 is hydrophilic. Once it is synthesized, it diffuses into the cytoplasm. It binds to the gated channel of calcium and causes the release of calcium ions. As we said also a few minutes ago, that protein kinase C is activated by DAG and calcium ions. This is a summary also of what we said here. For BIB2, who cleave it? Phospholipase C, thus forming IB3 and diacylglycerol. Exactly of this figure. But this figure gives you everything and every information that we need, as you can see. So this is the synthesis pathway for DAG and IB3. Now, just keep in mind that the IB3 function is releasing intracellular calcium ions, while DAG, its function is activation of protein kinase C. Now, IB3, as we said, what is the function of IB3? Releasing the intracellular calcium ions, which is normally stored in the smooth endoplasmic reticulum. It is gated channel. This is the gate. The gate is made up of 1, 2, 3, 4 subunits. For this gate to open, at least 3 IB3 must bind to 3 subunits to release. So the gate of the calcium ions on the smooth endoplasmic reticulum, the gate of these calcium ions is made up of how many subunits? 4 subunits. How many subunits must be activated by IB3 to be open? 3. Thus, the calcium is released. Normally calcium ions concentration in the cell is very low, 10 to 100 nanomole, very low. But once it is released into the cytoplasm, its concentration reaches from 500 to 1,000 nanomole. See how much increased. But this increase is for a very short time, because calcium is toxic for longer time. So calcium ions, once normal concentration inside the cell in the cytoplasm is 10 to 100, 10 to 100. Once its intracellular storage gated channel is open and the calcium is released, the intracellular concentration of calcium reaches 500 to 1000 nanomole. Now, calcium effect, calcium ions now as a second messenger also, we said. Calcium ions as a second messenger. We say always that calcium ions and other ions, they activate different proteins and enzymes. Do this calcium bind directly to the protein and enzymes and activate them? The answer is no. Calcium here binds to special binding proteins. For example, calmodulin. Then, at least 4 calcium binds to calmodulin and then activate it. Then calmodulin and calcium all together, they bind to different proteins, enzymes, or gated channel and activate it. So calcium ions does not bind alone and directly to the proteins and enzymes or gated channels. No, they must first bind to special binding proteins. For example, calmodulin. Then calmodulin binds to it 4 calcium ions to activate it. Then calmodulin, along with the 4 calcium ions all together, they bind to different enzymes and proteins and activate them. This is how calcium ions activate different proteins and enzymes. Now, we said that calcium ions are released from intracellular storage in the smooth endoplasmic reticulum. Now, from where this calcium comes? As we said, the first source is intracellular storage inside the cytoplasm, in the smooth endoplasmic reticulum. Also, some hormones, they bind directly to the calcium ions gated channel on the plasma membrane and causes the entrance of calcium ions. This is the second way. So the first way to increase the calcium inside the cells is by releasing the intracellular calcium ions. Second source is that the hormone bind directly to the receptors of calcium ions in the plasma membrane and causes the entrance of the extracellular calcium inside the cells. Third, also, the hormones indirectly, they cause depolarization of the cell membrane, thus causing the entrance of the calcium ions from outside of the cell. So, directly that the hormone bind to the gated channel of calcium ions. Indirectly, it doesn't bind to the gated channel, no. Indirectly, it causes the depolarization of the gated channels of calcium ions. So this is the source from where the calcium enter to inside the cell. IB3, as we said, also, how the cell turn off the IB3 signal by degradation to install by phosphatase. So phosphatase degraded or by phosphorylation to install. This is just an information direct one. Now, until now we studied cyclic AMP, cyclic GMP. We studied also the IB3 and calcium ions. Until now we studied 4 second messengers. The 5th second messenger is the DAG. The 5th is diacylglycerol. as a signal molecule. As we said, diacylglycerol from the name, it is made up of, as you can see here, this is the glycerol. CH2OH, CHOH, CH2OH. This is the glycerol, the black one. While in the yellow here is two fatty acids attached to a glycerol. This is why we call it diacylglycerol. Diacylglycerol. It functions as a second messenger. The diacylglycerol has two functions. The diacylglycerol has two functions. The first is releasing of arachidonic acid, the one you studied in the first year, that our cells use it to synthesize the prostaglandins. The second, as we studied also a few minutes ago, the function of diacylglycerol is stimulation of protein kinase C. So رجاء إغلاق المايكس كلها شباب، أرجو من الجميع إغلاق الميكروفون. So, as we said also a few minutes ago, that the function of diacylglycerol is stimulation of protein kinase C. Also, as we said, the protein kinase C is stimulated by calcium also. Another also molecule that stimulates protein kinase C, phosphatidylserine also. So this is the diacylglycerol. This is its structure and its function. With this, we come to the end of the second messengers and the G protein coupled receptors as a whole. So as we can see in this structure, that when the hormone binds to its receptor, just a summary of what we said, when the hormone binds to the receptor, to G protein coupled receptor, it causes what? A conformational change. This conformational change leads to activation of G protein and the replacement of JDB with JTB in the alpha subunits. Then the release of alpha subunits from beta and gamma that moves along the cell membrane. This until here, it happens to all G protein coupled receptor. What is the difference between one hormone and the other? Is the alpha subunit, what effector protein it activates. As we said, in cyclic AMP, for its cyclic AMP production, adenylyl cyclase. Cyclic GMP, guanylyl cyclase. For DAG, IB3 and calcium, what is the effector protein? Phospholipase C. Once the alpha subunit finds phospholipase C, it activated. What is the next step? Phospholipase C cleaves BIB2 into DAG and IB3. DAG then moves along the cell membrane until it finds protein kinase C and activate it. While IB3, because it's hydrophilic, it diffuses into the cytoplasm, then binds to the gated channel of intracellular storage of calcium in the smooth endoplasmic reticulum, causing the release of calcium ions. Calcium ions also can activate protein kinase C. And as we said, kinases, they phosphorylate different proteins and enzymes, as we studied in the last lecture. This is the IB3, DAG, and calcium ions. Now, we finished with the first example of plasma membrane receptor, which is the G protein coupled receptors. We said that most of the receptors on the plasma membrane belong to G protein coupled receptors. The second major types of receptors on the cell membrane is receptor tyrosine kinase. So G protein coupled receptors are the most popular, the most found receptors on the cell membrane. The second example that we will study is receptor tyrosine kinase. The second major type of receptors found in the cell membrane is receptor tyrosine kinase. Receptor tyrosine kinase, they are very important. They are, for our body, they are very important. Why? Because of their function. See their function. They regulate cell proliferation and differentiation. They also promote cell survival, see, and modulation of metabolism. Just concentrate on the regulation of cell proliferation. What is the cell proliferation? Cell division. If there is uncontrolled, or let us say it another way, if there is misregulation of RTK, receptor tyrosine kinase, shortly known as RTK. If there is misregulation of RTK, what will happen? What type of disease we or the people will get? Cancer. Many types of the cancers, whether in the lung, in the breast cancer, neolucus, or any and many other different types of cancers there is misregulation of RTKs in their body. So many cancers are caused by misregulation of RTK receptors. Why? Because we don't say why there is a mutation, because there are many reasons, but why in RTK? Because RTK regulates cell proliferation and differentiation. Thus, if there is misregulation of RTKs, then there will be misregulation of cell proliferation. And what is cancer? What is cancer? Uncontrolled cell division or cell proliferation. That's it. See how simple to define cancer. What is cancer? Uncontrolled cell division. See? Who controls cell division? The RTKs. If there is misregulation of RTKs, there will be cancer. Also, the misregulation of RTKs leads to diabetes, immune deficiency diseases. This is the most important diseases that resulted from misregulation. Misregulation means there will be a mutation or some defect in the receptor tyrosine kinase. Now, this is the structure of receptor tyrosine kinase. This is the structure. As we said before, for 2 lectures, the plasma membrane receptors in the last lecture. Any plasma membrane receptor is made up of 3 areas or 3 structures. The extracellular part, the cell membrane region, and the intracellular part or cytosolic part. Any cell membrane is made up of these 3 parts: the extracellular, the intracellular, and membrane spanning region, which is normally made up of what? Of alpha helix. What is the function of the extracellular part, the N-terminal? Here where the hormone binds. So always the hormone binds to the extracellular part, the N-terminal. The membrane spanning region is normally made up of alpha helix. While the intracellular part, the cytosolic domain, normally its function is to transmit the message of the hormone. So once the hormone binds to the extracellular part, it causes some changes in the intracellular part, that will transmit its message inside the cell. So this is the structure of tyrosine kinase. What is so special about, let us just remember a few points. What is so special about G protein coupled receptors? That it has two special things in the G protein coupled receptors. Its membrane spanning region is made up of 7 alpha helixes. What is the second special thing about G protein coupled receptors? That the G protein that binds to the intracellular part. Now, what is so special about tyrosine kinase receptor is that there is tyrosine kinase here in the cytosolic domain. There is tyrosine kinase here. And as we said, always, يا شباب رجاء إغلاق المايكات. What is the function of kinases? Phosphorylation, as we will see. So, why do we call it even the name tyrosine kinase receptor? Because in the intracellular part, there is tyrosine kinase protein, or we call it enzyme, as we will see. See here, one important information also. Dimerization of the tyrosine kinase receptors, or receptor tyrosine kinase, also we call it. The dimerization is a must for the activity of this receptor. So a single receptor is a monomer, we call it, is not active. There must be two receptors, they come together, we call it dimerization. So dimerization is a must for the activity of tyrosine kinase receptor. This is why tyrosine kinase receptors are found in two forms. Sometimes they are already dimerized or will dimerize once the hormone binds to them. So some receptor tyrosine kinase, they are found already dimerized. And others, the second type, will dimerize once the hormone binds to them. Thus, in any way, dimerization is a must. Dimerization is a must for the activity of tyrosine kinase receptor. See here. Now we will continue. Now, see how receptor tyrosine kinase act and concentrate here. See here. When the hormone binds to that receptor tyrosine kinase or tyrosine receptors, it causes their dimerization. This is the first step. So the hormone causes their dimerization. And if they are already dimerized, then the second step will start directly. Once the receptor are dimerized, then tyrosine kinase in the intracellular part is activated. See here and concentrate. Once the tyrosine kinase is activated, every single receptor, we call it monomer, every monomer receptor has, as we said, tyrosine kinase will phosphorylate the opposite monomer of the receptor. So this dimerization has two monomers, 1 and 2. If once they are dimerized, what will happen? Tyrosine kinase in the cytosolic part of this receptor will be activated. What is the function of tyrosine kinase? It will phosphorylate the opposite receptor. Which amino acid is phosphorylated? From the name, the amino acid tyrosine. because tyrosine kinase, it means it will phosphorylate which amino acid? Tyrosine. So is every monomer of the receptor will, so this monomer or this single receptor will phosphorylate a very specific tyrosine amino acids on the opposite of the opposite receptor. And same thing to this one, which the other one, which will phosphorylate a limited and specific number of tyrosine on the opposite receptor. This process, we call it autophosphorylation. What do we call it? Autophosphorylation. This phosphorylation of the tyrosine attract what we call them adapter protein. Thus, receptor tyrosine kinase, now after the hormone binds and there is autophosphorylation. This phosphorylated tyrosine amino acids in the intracellular part of the receptor, what will attract to them? Some adapter protein, for example, GRB2. GRB2, as you can see, also we call it a signaling molecule, has two domains like hands. One binds, once the SH2 domain binds with the phosphate, it activates SH3 also. The SH3, the other domain. So GRB2 has two domains, SH2 that binds with the phosphate and SH3 that activate the SOS, another signaling molecule. And once SOS is activated, it will activate another signaling molecule. We call it RAS. This RAS has JDB. Once it is activated, JDB is replaced by JTB. This we call it signaling molecule. Until these signalings also will affect the transcription of the DNA also. Many cancers also are caused by misregulation of RAS. RAS is very important signaling molecule. So, just to repeat very fast, dimerization is a must for the RTKase receptors. Thus, RTKs are found in two forms. They are already dimerized or well dimerized once the hormone binds to them. Anyway, dimerization is a must. Once the hormone binds to the receptors, if they are not dimerized, then they will dimerize. What is the second step? This binding will cause a conformational change. This conformational change will lead to the activation of which protein or enzyme? Protein tyrosine kinase. What is the effect of the activation of tyrosine kinase enzymes in the cytosolic domain of this receptor? Every monomer, every single receptor will phosphorylate a specific number of tyrosine amino acids. So this receptor, single receptor, will phosphorylate the opposite one, and the opposite one will phosphorylate the one that it's opposite also. In a process, we call it autophosphorylation. The phosphorylation of intracellular parts of tyrosine of this receptor attracts what? Adapter protein GRB2. GRB2 has two domains. It binds to phosphate through SH2 and activate the other signaling molecule SOS with SH3. Once SOS is activated, it activates another signaling molecule, which we call it the RAS. RAS has JDB. Once it's activated, the JDB is replaced by JTB. This is the signaling. This is the signaling. And it is a very wide, very wide science. and very active science, as we said also, because misregulation of these signaling molecules lead to cancers and different diseases. Now, signal termination. We finished with tyrosine kinase receptor. That's it about receptor tyrosine kinase. Now we will come to signal termination. How the signal of hormone is terminated. How the signal of the hormone has a signaling. It will signal as we said before, it will signal it will activate protein kinase A. Protein kinase A will phosphorylate some enzymes. Okay, how to end this phosphorylation? As we said before, phosphatase. We also said before that for termination of the hormone occur through degradation of the hormones by which organs? Mainly liver and kidney and the target cells also. But mainly who degrade the hormones? The kidney and liver. To less extent, the target cells. Also, the hydrolysis of JTB, because we said as long as JTB is bonded with alpha subunit, for example, it is active. If this JTB is hydrolyzed to JDB, the activity will be ceased. Another way for the termination of signals is by degradation of the second messenger, as we said. Phosphodiesterase, we said it converts the cyclic AMP into AMP, cyclic GMP to GMP, thus terminating the second messenger. So as we can see, there are many ways to terminate the signals of the hormones by degradation of the hormone, or by degradation or converting or seizing the second messenger, or by hydrolyzing the JTB, or by ending the phosphorylation by phosphatase. All of these causes signal termination. All of these causes signal termination and the end of the hormone signaling pathway. This is the last, this is the type of second messengers and what type of hormones that uses them, or what type of hormones that causes their release. And هنا المحاضرة الحمد لله. إذا عندكم أي سؤال تفضلوا.
دكتور أنا عندي سؤال.
تفضلي.
دكتور لما حكينا عن ال degradation of the second messenger بينهي ال signaling.
نعم.
طيب الهرمون لسه موجود يعني ما بيتكون second messenger ثاني؟
لا مو يعني ما بنحكي عن يعني ما بنحكي عن عملية واحدة فقط. نحكي عن كل هاي العمليات بتشتغل مع بعض، يعني ما في انك تنهي الهرمون وتخلي السكند ماسنجر، لعدا كده اذا عملتي ديجريديشن للهرمون شو بده يضله؟ بده يضله السكند ماسنجر. فاحنا ما بنحكي عن عمليه واحده بتشتغل لحالها، بنحكي عن انه كل هاي العمليات تشتغل مع بعضها for ending the activity of the hormones, for ceasing the activity of hormones. فلذلك كل هاي الباثويز تشتغل مع بعضها، ما في يعني كل هرمون او كل آلية تشتغل لحالها.
أوكي. تمام دكتور يعطيك العافية.
يا ميت مرحبا. إذا في أي سؤال؟
يعطيك العافية دكتور.
تفضل يا ميت مرحبا.
دكتور احنا حكينا بالمحاضرة انه الريجوليشن اوف كالسيوم يعتمد على ارتباطه بالكالموديولين، صحيح؟
نعم.
طب دكتور الكالموديولين only in the smooth muscle، يعني skeletal muscle يكون insufficient amount of calcium؟
لا بس هذا بيكون intracellular في معظم الخلايا. intracellular
مش
intracellular.
المصل الكالسيوم يرتبط مع تروبونين سي، ما يرتبط مع الكالمودولين دكتور.
لا كريجوليتر لازم يرتبط. ليش؟ للاكتيفيتي تاعت الكالسيوم. احنا حكينا انه كيف بده يرتبط مع العضله، او كيف بده يرتبط مع المايوسين اذا درستوها. النهايه بالنهايه انه الكالسيوم لا يمكن يرتبط مباشره مع اي بروتين او اي عضله او هذا الا مع وجود الريجوليتر بروتينز، ما بيرتبط مباشره. فلذلك يعني احنا بنحكي هون عن المولكيولر بيسس. ممكن اذا انت اخذتها باي موضوع ثاني انه يحكي لك انه الكالسيوم بيرتبط مع العضله او بيرتبط مع التروبونين او وات ايفر، بس في الحقيقه هو في البدايه بيرتبط مع هاي الريجولاتوري بروتينز. واحنا حكينا كمثال هو الريجولاتوري بروتينز هون يعني مش موجود، خلينا نحكي عنه. هنا كالمودولين احنا اخذناه كمثال فقط على السبيشال ريجولاتوري بروتينز، ولكن موجود منه يعني كريجولاتوري بروتينز موجود العديد من الانواع، ولكن الكالسيوم فور اتس اكتيفيتي لازم ترتبط مع هاي الريجولاتوري بروتينز. نعم. إذا في اي سؤال. شكرا لكم شباب، وشكرا على حسن استماعكم. بارك الله بيكم، وربنا يحفظكم ويحفظ أهاليكم جميعا. والسلام عليكم ورحمة الله وبركاته.